We leave this for the Professor Ashok Agarwal, the Director of the Center of Reproductive Medicine in the Cleveland Clinic in the United States of America. Professor Agarwal is a well-known researcher and scientist all over the world. He enriched our medical literature with hundreds of high-quality publications. Here, Professor Agarwal today will touch the concepts of proteomics and which are techniques to identify sperm-specific proteins, and will provide us with or will elaborate on the future of proteomics in male infertility. Please welcome Professor Agarwal.
Good afternoon, everyone here. First of all, I want to thank the organizing committee of the CGMR for their kind invitation. It has been a great pleasure for me to be here amongst all my friends and colleagues. We've been working for the last 3 years now, and this is my third or fourth visit, and this is the third meeting of the IGMC, so I'm very pleased to be here. I also want to take this moment to thank the chairperson, Professor Taha, as well as Dr. Gautam for chairing this session. So, what I wanted to do today was to talk about some of the research that we have been conducting in our center at Cleveland Clinic. And so my talk is going to be more about the work that we are conducting and just give you some of the latest findings, including some of the research that we have with our colleagues here at CGMR. So the things that we are going to cover are, so the learning objectives will be to talk about very briefly what are the limitations of semen analysis, discuss some of the new advances to evaluate the fertilizing capacity of the spermatozoa, discuss the proteomics and its role in infertility, some of our studies at our center on proteomics and male infertility, what are the challenges of conducting proteomics research with respect to male fertility, the limitations of introducing proteomics for male infertility patients as a diagnostic tool, and conclusions and some of the take-home messages. So this diagram is essentially showing the cellular changes which occur during the spermatogenesis or sperm production, and during the phase of spermatogonia to elongating spermatids are stages where transcription and repair occur before the sperm are released into the epidermis, and there are no more changes that occur once the sperm are released in the epidermis, and it is transcriptionally inactive or silent. So the incidence of male infertility is about 15% of all couples in reproductive age in the United States. And male factor contributes to about 50% of all cases of infertility. And in some parts of the world, especially in the Middle East, this could be as much as about 60 to 70% of the cases in a study that we published recently. In addition, about 25% of cases of male factor account for no identifiable cause and are called as unexplained infertility. So there is a limited amount of information. Semen analysis is one of the first tests that is used to evaluate a patient coming for male infertility workup. As a physician, Dr. Taha will tell that they will recommend 2 semen analysis 2 to 4 weeks apart. But it provides a limited amount of information because it only tells us about certain parameters that can be measured microscopically, such as count, motility, and morphology. But this certainly, I think, does not tell us much about the functional competence of the spermatozoa. So that is a major limitations of semen analysis. There are a number of advanced functional tests. Some of them are described here, many more are not. These are in no particular order: acrosome reaction test, a test for sperm capacitation, sperm membrane integrity, measurement of free radicals or reactive oxygen species, measurement of antioxidant capacity, measurement of sperm DNA fragmentation, lipid peroxidation assay. Some of these tests are being utilized by us in the Cleveland Clinic for routine testing after the semen analysis testing. So those are tests that can be used as an advanced test. So now talking about the proteomics, essentially the steps involved here are in the fragmentation of proteins into peptides, digestion of peptides and LCMS analysis, selection of proteins and by collision-induced dispersion, and selection by mass spectroscopy. So those are the things which involves proteomics. So the current focus is to understand the underlying molecular mechanism of sperm dysfunction. It is important to understand that because that would serve as a potential biomarkers in elucidating the causes of male infertility. So, why proteomics? Because the study of cellular components at the molecular level, it helps identify the differential expression of proteins which are either over or underexpressed or are present as a unique protein in a cohort of infertile men with various types of clinical diagnosis of fertile men. Sperm-specific proteins which help identify various metabolic pathways, processes or networks that are essential for the diagnosis of male infertility. And it also provides a greater understanding of protein function. Proteomics allows the identification of sperm-specific protein interaction associated with important steps in sperm metabolism. And for that, sperm is an ideal source because it's a fully functional, mature cell, translationally and transcriptionally inactive. And that's why proteomics is a better omics for studying this, unlike genomics or metabolomics. And it utilizes proteomic tools and bioinformatic analysis. The challenges in male infertility research is that advantages of using proteomics over routine semen analysis is that it confirms the qualitative and quantitative measures of a protein within the spermatozoa. However, identifying ideal biomarker for clinical diagnosis in a complex scenario to distinguish the pathophysiology is only one of the main challenges. Further, in a clinical proteomic study, one sample per individual will only give a snapshot of the disease. So it is really important that you need several samples to conduct a more thorough understanding of the proteins that are involved. So in longitudinal sampling, where samples are collected over a course of time to determine how the proteome changes over time within an individual is necessary to identify putative biomarkers. In a study that we published recently, we conducted a comparative analysis of protein in fertile and infertile men, and we ran it through DGel and conducted Marley-Toff. We found that we identified 4 unique proteins in the sperm of fertile and infertile men, which were seminal-2 precursor, prolactin-inducing proteins, cluster isoform, and prostate-specific antigen isoform. So this was the first study. And then in a second study that we published on 2-dimensional differential in gel electrophoresis, in this study we examined semen samples from 20 normal men with normal semen quality and 32 male infertility patients, divided them into ROS positive and ROS negative by conducting estimation of their seminal free radical content, and pooled sperm were used to prepare for 2DG analysis. and sperm were labeled with fluorescent dyes and excised protein spots. And we then ran the LCMS analysis. The main findings of this study was there were about 1,200 spots in patients with ROS positive and about 1,300 spots in ROS negative. And there were 31 of them were differentially expressed. Of these, 6 spot decreased and 25 spots increased in ROS negative samples compared with ROS positive. There were significant proteins with antioxidant properties seen in ROS negative sample. And this we concluded may explain the significance of ROS in the pathology of male infertility. This is a study that we published. Another study that we published was looking at the proteomic analysis of human sperm proteins with oxidative stress, which is an area of great interest to our program. In this study, we examined semen samples from 53 subjects, 20 donors, normal subjects, and 32 patients with male infertility. And here we again conducted the study, and we found that in this figure shows the percentage of proteins involved in various biological processes such as metabolic process, protein folding, protein regulation, reproduction, and response to stress too. And the dark blue bars here represent overexpressed protein, and orange bar the underexpressed protein in the ROS positive group. And this figure, it shows the cyclic AMP response element modulator of cramp signaling. Here the spermatozoa circled as 1 in red, and RODF1 and ACE1 are overexpressed, where those in blue, which is LDSC, is underexpressed in cramp pathway in ROS positive group compared to the ROS negative. So there were 74 proteins identified, and about 10 of them were overexpressed and 5 were underexpressed, and the number of proteins that we saw are listed here. The results suggest that these proteins can serve as a potential biomarkers of oxidative stress. This is the article that we published. And this is another study that we published where we looked at the proteomic analysis of seminal fluid from men exhibiting oxidative stress. And in this study, we examined the seminal plasma proteome in men with oxidative stress. The study population was the same. We looked at the seminal fluid, unlike the sperm proteins in this one. And we identified 124 proteins. 7 were common and 7 were unique proteins. And we identified proteins that protect against the oxidative stress. Men with oxidative stress have proteins that are underexpressed or oxidatively modified and contribute to male infertility. So in this diagram, we can see this figure shows the comparative analysis of differentially expressed proteins that were present in the ROS positive blue and ROS negative green bar, or were common to the 2 bars, which are orange. So the differential expression of these proteins in the cellular distribution are shown here in the biological processes in this particular segment, and in the third column are the molecular functions according to the differentially expressed proteins. So the main findings was that men exhibiting high levels of reactive oxygenated species show proteins that are either down-regulated or oxidatively modified, and we've identified KLK3 as a key protein involved in the majority of pathways, processes, and networks. This is the article that was published. There's another study that was published which looked at the differential proteomic profiling of sperm proteins in infertile men with unilateral or bilateral varicocele. And the main findings of this study were that we identified 253 proteins, 64 were uniquely expressed in the bilateral group and 31 in the unilateral varicocele group. Majority of these were involved in metabolism, apoptosis, and signal transduction. Core function proteins were post-translational modification, protein folding, free radical scavenging, etc. And the top pathways were protein ubiquitination and mitochondrial dysfunction. Again, our results provide insight into the underlying mechanism of varicocele-associated infertility in infertile men with unilateral or bilateral varicocele. This is another study which is in the press right now on the proteomic signatures of infertile men with clinical varicocele and their validation studies reveal mitochondrial dysfunction leading to infertility. Here we examine the protein identified from our varicocele studies on unilateral, bilateral, and comparison between unilateral and bilateral varicocele groups. A total of 50 subjects were enrolled, and what we found here is, this is the Venn diagram here, which shows a total of 1250 proteins. 138 were unique to fertile control group, and 140 of them were unique to unilateral varicocele. 120 were unique to bilateral varicocele group. And the Western blot analysis was done to validate our results. We selected 5 proteins for validation, and these were based on their role in stress response and regulation of structural and functional integrity. And these were the down-regulated under-expressed protein were PKAR1, which is cyclic AMP-dependent protein kinase protein, AK7 protein adenylate kinase, CCT6B, which is the T-complex protein 1, and heat shock protein. Upregulated or overexpressed protein were ODF2 or outer dense fiber protein 2. So the overall summary of various studies that we discussed and many more that have been published recently from our program shows that sperm proteomics is an important tool in understanding the basic underlying mechanism of male infertility. Various type of technique such as 1D, 2D or 2DG can be used prior to LCMS analysis, and proteomic analysis and bioinformatic tools are important in identifying the differentially expressed proteins. The differentially expressed protein alteration is responsible for various reproductive functions which are related to male infertility, and proteins of interest can be examined further as potential biomarkers of male infertility. So the future of proteomic study in terms of the goal is to fully characterize the human sperm proteomes. This is a big job which will take some time, but there are thousands and thousands of proteins. Understand differences between various stages of spermatozoa, identify post-translational modification that will lead to suboptimal sperm quality, develop novel diagnostic tools and therapeutic drugs to improve sperm dysfunction, apply sperm proteomics not only as a research method but also as a clinical diagnostic tool in the field of male infertility. These are my colleagues from Cleveland Clinic, Dr. Rakesh Sharma, Dr. Damayanti from now in Kuala Lumpur, Malaysia, Ahmed Ayaz from Istanbul, Luna Samanta from India, Ricardo Bertola from Sao Paulo, Gulfam Ahmed from Pakistan, and from CGMR, Dr. Murad Asidi, Mohammed Abu Al-Magad, Dr. Adem, and Rola, Turkey. And these are some of the studies which were published. Some of them are listed here, some are not, which were in collaboration with our colleagues from CGMR. We are also looking for a candidate to pursue this research in our program. If anybody is interested, they can reach out to me, and we'll be happy to talk about that. And these are some of my colleagues in the Cleveland Clinic. Thank you very much.
Thank you very much, Professor Agarwal. Very stimulating presentation. The floor is open for questions and comments.
We are quite far. I think it's a very promising area, but the main challenges of proteomics for diagnostic purposes in infertility cases is essentially it is a very sophisticated technique. It requires a great amount of expertise, expensive instrumentation, and it is it is a very labor-oriented technology. The validation is very important because the results are not easily reproducible. It requires immense amount of standardization to achieve something which can be repeated by the same investigators or by other investigators. So I think it definitely is a technique which is very promising as something that may happen, but I believe for another maybe 5 or 10 years, maybe sooner, but it doesn't seem like we have a lot to discover, especially in the spermatozoa proteomics field. We have just started, so it's basically like just the beginning of the whole unraveling of the whole sperm proteomes. There is a lot of work to be done.
Ashok, thank you very much for the talk. Doing the proteomic profiling during spermatogenesis is very important. Like doing it for the spermatogonia, primary spermatocytes, secondary and spermatids. How you would dissect that?
How do you
dissect the cellular level?
Really, we are not, except for some of the studies where we did separate out the mature and immature sperm using the double density gradient, actually 3-layer density gradient, where we were able to separate the spermatozoa which are mature versus immature spermatozoa or white blood cells. There isn't an easy method to separate these cells in enough quantity for proteomic analysis, because you need enough amount of proteins. So separation is just one part of the puzzle, but the biggest problem is to have enough number of cells then to use it for downstream processes, that you require enough number of proteins not only to run the proteomics but also to do the validation studies on the same samples later on. So it's very difficult.
Dissecting it as the mature and immature, quite general, isn't it?
Yeah.
My question is, are you going to dissect it stage by stage, or that would be really difficult?
It will be, at least in humans, is very difficult. I think there isn't any such type of technology right now which will allow us to do it in a way which will allow enough amount of cells. You might be able to do it, but the number of cells will be too small, and then it may not be very pure. It will not be very pure population of those cells. So the purity of the cell population will be compromised.
Any more questions? Okay.
Thank you, Ashok. Very nice talk. As usual, my question is regarding the intra-patient variation and intra-group variation between these data, especially about the number of samples limiting and the amount of as well.
Yeah, those are genuine concerns, and there is a way that you can get around these things, and I think there are biological variations as well as sample variations and technical variations that have to be taken into account. So for some of the things you can run the samples in triplicate to avoid the variation, especially the technical variation. For biological variations, you can pool the samples and put them all together in a group of samples so that you can reduce the variability as well. So those type of issues have been studied and reported in the literature quite well now, and I think most of the people who are working in the proteomics field are able to conduct these studies quite well.
One last question. Okay.
My question concerns ROS. Is it the cause of some genetic defects in the patients, or it is the effect?
Well, it's the chicken and egg type of situation, which came first. We know that there is a strong association. I think many of you are geneticists here, so you might be able to tell more. But we know that that reactive oxygen species, or ROS, is an underlying cause of sperm dysfunction through the process of lipid peroxidation. So it causes cell membrane breakdown and causes a variety of damage leading up to the cell DNA damage. So those type of changes are already known and have been published in hundreds of articles. But its direct relationship to genetic changes further than DNA are not studied in experimental setting. I think most of those studies are, association studies that have been published. So I really don't have a clear answer in terms of the impact on genetic.
Okay. I have a question, Professor Agarwal. If you find a defective protein, if there is any chance to fix or rectify this defective protein using proteomic techniques?
That is hypothetical at this time. We are at a very basic stage of mapping out all the proteins under different clinical, diagnosis of male infertility, such as varicocele or azoospermia. And so, certainly, I think this is a good question because as a clinician, you would like to know. We will probably think that it is possible if we can correct the underlying cause that is behind the problem with the sperm dysfunction and the protein that may have been the cause of that. So if the underlying cause can be corrected, it is possible that that protein perhaps will change its function. And maybe instead of being underexpressed, it might be normally expressed in those sperm samples. So it is theoretically possible.
Okay. I hope we wouldn't run out of time, but I have a last question.
Sure.
You looked at the sperm-specific proteins in the ejaculate only. Did you look at the testicular tissue taken by biopsies?
Not really. I think these are difficult because the amount of testicular tissue that we get during the biopsy is very small. And to conduct the proteomic analysis, you require a decent amount of tissue, because those tissue that we get, we use it to prepare it for IVF or ICSI. So there isn't enough tissue left for conducting the research. And especially in humans, it is very difficult. If you want to conduct research in animal, that it may be possible, but certainly with the sperm, it is easier because you have a large number of cells in many patients. But testicular tissue amount is very small. You cannot take out a whole testis and give it for research.
Okay. Thank you very much, Prof.